itu ada dua spelling. So on your chapter 14 notes, okay, ini succinate dehydrogenase, that's complex 2, alright? Dia ada terlibat dalam oxidize succinate pergi fumarate. Dia ada terlibat dekat step itu. So inilah target complex for malonate. Macam Jordan tanya tadi, cyanide hanya target complex 4 ke? Yes. Whereas for malonate, dia hanya target complex 2. Dia compete dengan succinate. Ini masa Krebs cycle, okay? During Krebs cycle, dia mungkin ada melibatkan enzim ini untuk oxidize succinate to become fumarate. So, ada malonate ini sebagai competitive inhibitor untuk compete with succinate. Ini tukar ya, sebelum ni Miss letak succinic. So, please change it to succinate dehydrogenase. Okay. And now let's look at the last part of chapter 15. This one, over here, I think we would just cancel off anaerobic respiration lah. Tukar title dia, tak perlu tulis anaerobic, terus fermentation sahaja. Please correct this, it's just fermentation. Kita tak nak sentuh anaerobic, which is different. Sudah?
Sudah, Miss.
Okay. Right. So fermentation is a very, very short part in this whole chapter. Bahagian ni memang pendek je dan you dah pernah belajar masa secondary school, so it's not really new to you. First of all, you will have to remember the definition of fermentation. It is the incomplete oxidation of glucose to carbon dioxide, ATP, and ethanol kalau ini ialah plant cells. Kalau plant cells or yeast, then we will have carbon dioxide, ethanol. Whereas if it's animal cells, we will have lactate and ATP. Tak ada carbon dioxide, okay? No carbon dioxide untuk yang in the animal. So when you write the definition, you kena baca soalan betul-betul. Whether fermentation itu berlaku dalam plant cells ke, yeast cells ke, ataupun soalan tu dah bagi tahu is in the muscle cell. Kalau dia dah cakap muscle cell, then your definition of the fermentation should be the incomplete oxidation of glucose to lactate and ATP in the absence of oxygen. Jangan tulis dua-dua ya. So rujuk soalan. Dia tanya pasal plant ke atau animal. Kalau dia dah specify muscle cells, itu mesti ialah animal, then you just give the definition for animal cells. Can you understand?
Boleh, Miss.
Boleh, Miss.
Right.
Yes, Miss.
Kenapa Miss nak Q cancel off anaerobic? Sebab anaerobic ini complete oxidation. So dia tak ngam dengan definition ini. Definition ini, when we talk about incomplete, it's only for fermentation sahaja. So anaerobic dengan aerobic, dua-dua pun complete oxidation. Hanya fermentation je yang melibatkan partially breakdown, partially oxidize of the glucose in the absence of oxygen. Dia hanya partially breakdown. Because it's only partially breakdown or partially oxidized, only a small amount of energy is produced. Dia hanya dapat produce ATP semasa glycolysis sahaja. Apparently, if it's during glycolysis, then only 2 net ATP is produced. Okay, so what happened with fermentation? Konsep dia sangat senang. Konsep untuk fermentation ialah first start dengan glycolysis, next reduction, tu je. That's all. Fermentation is all about glycolysis. That's when it produces the ATP. And then terus reduction. Tu je. It's as simple and straightforward as that. And now, again, we will have to remember the role of NAD plus as the oxidizing agent and NADH as the reducing agent. In this case, it does not involve Krebs cycle and oxidative phosphorylation. Tak ada link reaction, tak ada Krebs cycle, tak ada oxidative phosphorylation, semua tak ada. Dia hanya ada glycolysis sahaja. So, class, what do you think? The production of ATP, dah bagi dekat sini, sorry. The production of ATP is only through substrate level phosphorylation sahaja. Tak boleh melalui oksidatif lah sebab dia pun tak masuk mitochondria, dia pun tak ada stage yang seterusnya, dia hanya first stage tu je. Lepas glycolysis terus reduction. Yang stage seterusnya semua tak ada, so maksudnya ATP dia hanya boleh produce melalui substrate level phosphorylation semasa glycolysis. Jelas?
Yes, Miss.
Jelas,
Okay.
Miss.
Alright, so inilah yang you kena ingat, Miss dah letak star. You must remember all this. Kalau you ingat ini, you memang dah faham lah fermentation. Fermentation, there are two types. Alcohol dengan lactic acid or lactate. Ini nama product form. Okay? Nama fermentation ini dia ikut nama produk dia. What does that mean? If you look at page 11, nota muka surat 11, slide ketiga, ada jadual ini. If you look at this table, then you can easily tell lactate fermentation products dia lactate. Alcohol fermentation products dia ethanol. Okay, and ingatlah ada O, co, so carbon dioxide. Dia je yang ada carbon dioxide. Lactate tak ada. So what do you think? Kalau dia ada carbon dioxide release as a byproduct, what process will you expect?
Decarboxylation. Decarboxylation.
Very good. Whereas this one does not involve decarboxylation. That's why in the products, we do not have carbon dioxide form. Ingat alcohol, carbon dioxide. So ethanol, dia ada carbon dioxide, that means decarboxylation is included, dia ada involved. And tadi kita cakap dari glycolysis dia terus sambungkan fermentation. So what do you think? During glycolysis, one molecule of glucose akan jadi two molecules of
Pyruvate.
very good. Pyruvate, how many carbon?
3.
Very good. So now what do you think? Kalau pyruvate dia ada decarboxylation. So maksudnya ethanol ini 2 carbon ke 3 carbon?
2 carbon.
2 carbon. Tapi you kena tahu sebelum dia jadi ethanol, dia akan jadi satu intermediate dulu yang kita panggil ethanol atau acetaldehyde. Dia bukan terus jadi ethanol. Tak. Dia akan jadi satu intermediate dulu, baru dia jadi ethanol, which is the alcohol. Ingatlah the last one mesti OL sebab dia ikut nama fermentation, alcohol. So the last one is ethanol. So ethanol apparently is 2 carbon sebab decarboxylation sudah berlaku. Okay, so that's for alcohol fermentation. Whereas if it's for lactate fermentation, ada tak melibatkan decarboxylation? Ada decarboxylation ke?
Ada.
Tak ada. So maksudnya lactate or lactic acid ini 3 carbon ke 4 carbon? 3 carbon ke 2 carbon?
3.
3. It's important to know this because in the exam they may ask you to write the equation. Sebab tu dalam nota Miss ada bagi equation. Ada satu tahun tu kami tak expect pun dia perlukan student hafal. All this time we just teach them, okay, you just have to know the products name, it's fine. Tak perlu ingat equation. Tiba-tiba satu tahun tu dia keluar suruh student tulis equation, 2 markah. That's why it's important to know the number of carbon, so it's easy for you to remember the equation. Okay, and that's it for lactate fermentation dengan alcohol fermentation. I will come back to this to talk about final electron acceptor. Nanti lepas kita tengok dua-dua itu, I will come back to this. Now, how to get the name of the fermentation is based on the end product. Kalau end product ialah lactate, then the name lactate fermentation. Kalau end product ialah ethanol, then it is alcohol fermentation. And what about the similarity? Since both only involve glycolysis, it only occurs in the cytoplasm. And both in the absence of oxygen. Sebab hanya melibatkan glycolysis, so hanya produces 2 ATP. I tak tahu boleh ke tak kalau you tulis both involve glycolysis. Supposedly boleh, tapi saya buat jadual ini ikut past year answer scheme. Mungkin dia tak terima ke, dia tak masukkan ke. So, ikut utamakan 3 points ini dululah. Kalau you nak tulis essay, tulis lebih, masukkan je. Both involves glycolysis if you want as an extra point. But please make sure you write this 3 first as the similarity. If you want more, then you can write both involve glycolysis if you want. Any question?
No, miss.
Right. Okay. So like I mentioned just now, lactate fermentation is in animal cell or muscle cell. Alcohol pula dekat plant cell, yeast cell or bacterial cell. Ada je bacterial cell pun carry out lactate fermentation. Ada But this one, we put it here, alcohol fermentation. You should remember for yogurt. Nanti Miss akan ajar untuk yogurt, ada je bacteria yang terlibat dalam lactate fermentation. Anyway, lactate fermentation kita selalu guna contoh animal cell. Most of the time in the past year question pun dia tulis animal cell sebagai contoh. Now let's look at alcohol fermentation. Am I too fast?
Boleh juga, miss.
Boleh ya. So apa yang Miss letak star ini kena tahulah. So first of all, cancel the title. Sebab I takut you keliru. So cancel it, tak nak tulis anaerobic, terus fermentation. So fermentation kita ada 2, satu alcohol, satu ialah lactate fermentation. Under anaerobic conditions, in the absence of oxygen, plant cells or yeast or bacterial cells will undergo alcohol fermentation. ATP produced is just 2. I know you may ask, Miss, bukan 4 ke? Miss cakap kalau dia cakap total ATP tu 4. Right, over here, in the part for fermentation, dia macam tak emphasize sangat mesti 2 net ATP. Dia tak. Kalau fermentation part, you tulis 2 ATP, dia pun terima. It's just for glycolysis je dia sangat, I mean this is also glycolysis, tapi kalau dia dalam bahagian fermentation, it's not aerobic respiration, dia tak straight sangat. You tulis 2 ATP, dia pun bagi betul. Tapi kalau soalan glycolysis untuk aerobic respiration, mesti berhati-hatilah. How many ATP? 4. How many net ATP? 2. But when it comes to fermentation, you tulis 2 je pun bagi betul. I hope I didn't make you confused because I can be quite confused too when it comes to the question. Kadang-kadang tak tahu apa yang dia nak. But over here, fermentation, memang all fermentation kita hanya cakap 2 ATP. you tak mention to net pun tak apa untuk bahagian fermentation. Are you okay?
Okay, Miss.
Okay. Selain daripada Iwa, orang lain boleh?
Okay,
Boleh,
Boleh,
Miss.
Miss.
Boleh ya? Okay.
Okay, Miss.
Right. So this one is quite easy to understand, to be honest. Let's see. Okay, kita tengok yang Miss lukis lah. Okay, first of all, kita start dengan glucose. Dia mesti start dengan glucose. So let's start with alcohol fermentation first, which happens in the plant cells. Yeast means yang dalam roti itulah alcohol fermentation. And then, or some bacterial cells. All right. So the first one mesti glucose. And then from glucose, during glycolysis, dia akan form pyruvate. During glycolysis, kita ada oxidizing agent apa? NAD plus ke FAD?
NAD plus.
Very good. NAD plus sebagai oxidizing agent. So NAD plus pula sendiri akan jadi NADH. Now, alcohol fermentation, alcohol dekat sini ialah ethanol. Ethanol ialah 2 karbon, tapi pyruvate ialah 3 karbon. So maksudnya, untuk jadi 2 karbon, dia melibatkan,
Decarboxylation.
good job, decarboxylation, whereby in this stage, in this process, carbon will be carboxyl group dia buang jadi carbon dioxide. So carbon dioxide will be released as one of the byproducts. And now from pyruvate dia akan jadi 2 carbon compound which is ethanol dulu. Ataupun kita panggil acetaldehyde. You tulis mana-mana pun betul. Dalam exam kalau you tulis ethanol pun betul. Tapi pastikan you tahulah nama lain dia acetaldehyde. Jangan nanti orang bagi acetaldehyde, you're like, what is this? So ingatlah itu acetaldehyde, boleh tambah dalam nota. Ethanol. Sebab dalam nota yang gambar ni Miss bagi acetaldehyde, so you tambah sendirilah itu ethanol. I ada bagi dekat sini juga. So far, so good. Alright. And now, after glycolysis, it's, yeah. Eva ada soalan? Oh, ada. Semua tak ada line ke sekarang? Atau
Mau hilang sudah, Miss.
mau hilang sudah?
Katak, Miss.
Tidak
Apa?
tidak elektrik, Miss.
Iya. Iya, mati.
Miss sambung je, okay?
Okay, sudah balik.
Mula hilang sir. Ritual! Yalah, sir pun boleh lah. Okay, anyway, I will just continue. I will just continue so that nanti you boleh tengok balik recording ini Sir Lim. Boleh lah itu. Nanti Miss ajar Koko sudah. Okay, so tadi Miss ada bagi tahu fermentation is all about glycolysis and reduction. So tadi kita dah cakap pasal glycolysis. So the next part, it must be reduction. Now, reduction, dia mesti perlukan reducing agent. Reduction of ethanol. Kita perlukan reducing agent. So, reducing agent dekat sini ialah NADH. So, inilah reducing agent. Reducing agent NADH, it will be used to reduce the ethanol or acetaldehyde. So, dia sendiri pula dah jadi NAD+. dia mesti ada proses ini. Kalau tak ada, dia tak dapat recycle NAD+, tak ada NAD+, glycolysis tak boleh continue. That's the reason why it's very important to have NAD+. Okay. Now, after reduction of ethanol using NADH, barulah dia jadi ethanol, which is the final product. That's the reason why we have carbon dioxide and ethanol as well as ATP, semasa glycolysis, as the products for alcohol fermentation. Yang ada line boleh faham?
Ada salah, Miss. Ada sudah electric.
Ada sudah. Okay.
Boleh.
Boleh ya? Kesiannya weh. Nanti pula UPS. Do I have to repeat on this part?
Iya, Miss.
Okay. Right. Iya. Okay, so tadi Miss dah cakap semasa fermentation mesti ada dua processes. Okay, early when we start, I cakap fermentation mesti melibatkan glycolysis untuk produce ATP as well as ada reduction. So kalau dah lepas glycolysis, dia akan sambung dengan reduction. And when you look at this one, mesti start dengan glucose. Sebab glycolysis kita mesti start with glucose. Glucose undergo glycolysis jadi pyruvate. So semasa glycolysis itulah yang dia akan produce ATP melalui substrate level phosphorylation. And semasa glycolysis juga yang dia akan ada NAD+ to be used as oxidizing agent. And then jadi NADH. Bahagian ini boleh faham? Bahagian glycolysis.
Boleh, Miss.
Okay, sudah lah kan? But then, if you look at the product, ethanol is 2 carbon, but pyruvate is 3 carbon. That's why you can expect decarboxylation berlaku. From pyruvate, which is 3 carbon, it will undergo decarboxylation to become 2 carbon. Tapi sebelum dia boleh jadi ethanol, dia akan jadi satu intermediate dulu, which is ethanol or acetaldehyde. Dia kena jadi ethanol dulu, barulah dia undergo reduction, jadi ethanol. Reduction ini reduction siapa? Reduction ethanol. Reduction of ethanol. By what? By the reducing agent NAD+. Maksud NADH. Maksudnya NADH buatkan ethanol reduce. And then ethanol dah reduce jadi ethanol. Itulah final product, which is also 2 carbon. So the equation here, you will have glucose, which is C6H12O6, C6H12O6, jadi ada carbon dioxide, ada ethanol yang 2 carbon. So 2 carbon, C2H5OH. Kena hafal ya. At least you tahu dia 2 carbon. Plus 2 ATP. Inilah equation dia. Ada ethanol, ada carbon dioxide, ada ATP. Balance it. Any question on alcohol fermentation?
Okay, Miss.
Dah balik? Alright. So that's it for alcohol fermentation. Apa yang you kena ingat dekat sini? Equation ini, products dia, carbon dioxide, ethanol, 2 ATP, and how it occurs. Ada decarboxylation, ada glycolysis dulu, and then ada decarboxylation, and then ada reduction. Done. So now, the question may ask you, what is the final electron acceptor here? Okay, the final
Oxygen.
electron... Itu aerobic respiration. What about the final electron acceptor for alcohol fermentation? Right. Final
NAD plus H.
NADH ialah reducing agent. Right, class. Reduction means gain ke loss electron.
Gain.
Reduction. Okay. Reduction maksudnya gain electron.
Gain.
So, kalau I cakap reduction of ethanol, maksudnya siapa yang gain electron? Reduction of ethanol. So,
NAD,
apa yang
NAD plus.
bukan. Reduction of ethanol.
Ethanol.
So, apa yang gain electron?
Ethanol.
Ethanol lah. Ethanol yang reduce. Maksudnya ethanol yang gain electron. Maksudnya ethanol lah yang final electron acceptor. Accept dari mana? Elektron ni dari mana? Elektron ini dari NADH. Sebab tengok NADH nanti jadi NAD+. Maksudnya dia dah hilang. Dia dah hilang elektron. Make sense? Elektron yang hilang ini dah pergi mana? Dia dah bagi dekat ethanol. Boleh faham?
Boleh, Miss, boleh. Boleh, Miss.
Boleh, Miss.
Okay, again, I will explain this. Right. Dekat sini, soalan ada je pernah tanya about final electron acceptor. Of course, when we say aerobic respiration, kalaulah masa kita belajar aerobic respiration, the final electron acceptor is the oxygen. Sebab dia lah yang terima elektron. So dia yang last terima elektron, so dia final electron acceptor. But when we talk about the final electron acceptor in alcohol fermentation, kita kena tengok berhati-hati ya. Alcohol fermentation, apa yang last terima elektron? First, you kena tahu elektron tu dari mana. NADH sebagai reducing agent, you nampak dia nanti jadi NAD+. Maksudnya dia dah hilang H and electron. Elektron ni dah hilang. Hilang pergi mana? Hilang pergi ethanol. Sebab ethanol reduce. Sebab ethanol gain electron. So, ethanol ini terima elektron dari NADH. Dia terima, maksudnya dia electron acceptor. Make sense?
Faham, Miss?
Faham eh? So dia yang last terima elektron. So dia lah final electron acceptor dalam alcohol fermentation. So first you kena ingat dulu, elektron tu dari mana? NADH nanti jadi balik NAD+, mestilah dia dah hilang elektron. So elektron tu dah pergi mana? Dia tolong reduce ethanol, maksudnya dia bagi dekat ethanol. Ethanol reduce, ethanol gain the electron from NADH. So, dia final electron acceptor in alcohol fermentation. That's for alcohol fermentation. What about lactate fermentation? Lactate fermentation sangat-sangat senang. Very straightforward. Okay, now let's talk about lactate fermentation. Berlaku dekat mana? Animal ke plant?
Animal.
Animal. So, animal cell or muscle cell. Alright. First, kita mesti start dengan apa? Kalau glycolysis, kita start dengan
Glukosa.
glucosa. Okay, glucose. Glucose. Macam Korean pula. Glucose. So, glucose. Glycolysis akan jadi
Pyruvate midah.
Pyruvate.
pyruvate. Pyruvate. Korean sangat. So, dia akan produce ATP lah semasa glycolysis. Okay, done. During glycolysis juga kita ada guna NAD+ sebagai oxidizing agent And then dia akan jadi NADH. Right. So far, so good.
Boleh, Miss.
Boleh ya? Nama pun dah lah lactate fermentation. What is the end product here?
Lactate.
Lactate. Tu je. Lactate atau lactic acid. Okay. So pyruvate nak jadi lactate. Glycolysis. Lepas tu mesti reduction. Tu je. Now, reduction of what? Reduction of siapa yang reduce? Pyruvate ke lactate? Pyruvate, very good. So reduction of pyruvate. So reduction of pyruvate, we use what as the reducing agent?
NADH.
Very good. Reducing agent ialah NADH. So NADH akan jadi NAD+. Again, you can see that NADH jadi NAD+, maksudnya dia pun hilang elektron. Elektron tu dah bagi dekat apa?
Pyruvate.
Dah bagi pyruvate. That's why we say reduction of pyruvate. Sebab reduction of pyruvate, it means pyruvate ini gain electron from the NADH. So, dia yang terima elektron. Dia lah final electron acceptor. That's all about lactate fermentation. Lepas glycolysis, reduction. Tu je. Sebab ini 3 carbon, ini pun 3 carbon. So, tak ada decarboxylation, tak leceh macam alkohol. Alkohol ini kena decarboxylation dulu, baru reduction. Ini tak. Lepas glycolysis, terus reduction. And you have to remember the equation. So, glucose mesti ialah C6H12O6. Then dia jadi lactate. 3 carbon. Terus ingat, 3 carbon. And then you have to memorize H6O3 plus 2 ATP. Balance it. So 2. That's all for lactate fermentation. Any question?
Miss,
Hmm?
jadi yang glukos tu dia produce satu lactate lah kan? Dua.
2.
Oh, okay.
Sebab ini 2 pyruvate, so 2 lah. Sebab 3 carbon je kan, dia 6 carbon. So it will be 2 lactate. Okay. Okay. Now, why do we need lactate fermentation in the muscle cell? Imagine in class, when you run kan you sampai tercungap-cungap. You macam tak cukup oksigen. But then you still want to run. Tak cukup oksigen, tapi you perlukan energy untuk run. That's when lactate fermentation is important. What for? To ensure ATP production continues even in the absence of oxygen. Even though masa kita lari tak cukup oksigen, tapi kita perlukan energy, at least ada lactate fermentation tolong produce the ATP untuk sementara, okay? To make sure that the ATP can still be produced, even though it's not a lot, only 2 ATP produced. But then, at least ada energy ini untuk keep the cell working. And that's the importance of lactate fermentation in our muscle cell. To make sure we can still have the energy needed for the process, even we don't have oxygen. Did I make it clear?
Yes, Miss.
That's what I wrote here. During vigorous exercises, maybe you nak lari kena kejar anjing ke, then you were running and then you need a lot of energy to keep you running, but you don't have enough oxygen. So it's fine because our muscle cells can still produce ATP even when we don't have oxygen. So the cells will turn to lactate fermentation and produces a little amount of ATP, only 2 ATP produced, but it's enough to keep the cells working. And that's the importance, and ini pernah keluar dalam past year. So, ini untuk explanation pernah keluar dalam essay 10 markah. So dia nak you bagi tahu dulu the details of glycolysis. So sini sampai sini pun glycolysis. Ini je lactate fermentation. Sebab lactate fermentation hanya satu step je. From pyruvate reduced by NADH to lactic acid. That's all. Okay. I don't need to go through this, right? You all boleh faham sudah kan? Glycolysis. Nak muntah sudah berapa kali kami repeat. Okay ah?
Okay, Miss.
Okay. Right, the last slide. We are almost done, okay? Last slide ini je, then we are done. Importance of fermentation in industry, you have to remember these examples, okay? They may ask you to give 2 importance of fermentation, give 3 importance of fermentation dalam structure. So you kena tahu macam mana, what are the importance. Bread making fermented food such as tapai, tempeh, thosai, kimchi pun boleh. Okay, and then making dairy products such as yogurt, cheese, manufacturing beer, wine, alcohol beverages. And ini kena ingat lah, ingat contoh importance. Now bread making, they may ask you, why is yeast used in bread making? Tadi kita cakap yeast terlibat dalam lactate ke alcohol fermentation?
Alcohol.
Dah bagi tahu, sorry ya. So yeast will undergo alcohol fermentation, and during alcohol fermentation, you can see that carbon dioxide is released. Sebab ada carbon dioxide, itu yang buatkan our bread fluffy. Kalau you makan roti, next time you boleh nampak, kenapa dalam tu ada lubang-lubang? Itulah sebab ada carbon dioxide produced. The air produced in the bread will make the dough rise. Make the bread dough rise. Dia buatkan tepung tu, dia akan, dough tu ialah tepung kan? I mean the bread dough. How should I
Yeah,
explain?
Miss. Yeah.
Yeah. Miss punya Malay memang limited edition, okay? The bread dough. This is the dough. Okay? Ni lah dough. Tengok. Okay, so to make it fluffy, we add in yeast. Why do we need to add in yeast? Nah, nampak ada lubang. Lubang ini disebabkan oleh
Udara.
sebab apa?
Carbon dioxide.
Carbon dioxide. Sebab carbon dioxide ini produced during what process?
Decarboxylation.
Alcohol fermentation bah. Bukan decarboxylation. Carbon dioxide ini produced disebabkan oleh alcohol fermentation. During alcohol fermentation, dia ada release carbon dioxide. That's how you answer it. Bukan decarboxylation. So you have to answer it as yeast undergo alcohol fermentation in the absence of oxygen. Semasa inilah alcohol fermentation yang dia produce carbon dioxide. Untuk apa? Untuk buatkan the bread dough to rise. Make sure you jawab ikut cara ini. Tak boleh terus cakap decarboxylation. Decarboxylation only this part. But what process involve decarboxylation? It is the alcohol fermentation that happens in the absence of oxygen. Tolong faham cara untuk
Aja, Miss.
Okay.
Okay, Miss.
And now, wine is made from grapes. Beer is from the maltose. Um, it depends. Ada je beer yang buat daripada wheat, ada beer yang dari barley. So it depends. Wine tu memang from grapes. Either yang purple tu atau yang green. Kalau red wine tu yang grapes yang anggur yang purple. Okay. Anyway, making dairy products such as yogurt. Ini you kena ingat kenapa yogurt jadi solid. Kenapa yogurt tasted sour. Lactate is lactic acid. Okay, you have to remember that lactate ialah acid. Acid maksudnya dia lower pH. So kalau lower pH, mestilah dia akan jadi protein tu coagulate sebab denaturation. If you still remember, low pH will cause denaturation of protein. So protein tu jadi coagulate, solid. And itulah dia form yogurt. And why does it taste sour? Sebab lactate acid. Acid tu sour. Itu je. If you next time buy yogurt, when you read the ingredient, you may find the type of bacteria that we call lactobacillus. So lactobacillus, what shape?
Fruit shape.
Okay, good job. So lactobacillus is the type of bacteria that will undergo lactate fermentation. So ada lactic acid, ingat je acid, low pH, sour. So ini yang you kena ingat. And that's all for today. Oh, ini you boleh baca sendiri ya. Okay. Extra information. Lactate ini nanti macam mana kita nak get rid the lactate in our muscle cell. You ada muscle cramp because the lactic acid accumulate in your muscle cell. Itu yang buatkan the cramp. So how to get rid of this lactate over time? They will send to the liver, and then dekat liver itulah yang dia boleh remove the lactate. Okay, we are done. So any question?
No, miss. Yay.
Ini baca juga ya, sebab I tak tahu dia akan tanya dalam exam ke tak. So that you know, lactate tu dia tak stay je dekat muscle cell. When we have oxygen again, dia boleh send this lactate from the muscle cell to the liver, and dekat liver yang dia akan jadi balik pyruvate. And that will reduce the cramp. Soalan kita bincang next week. Alright, soalan untuk fermentation, we'll discuss them next week. Before I end the class, do you have any question on fermentation?
Ya dah, Miss.
Boleh faham sudah final electron acceptor ini? Okay?
Faham, Miss.
Stop boleh lah, Miss.
Boleh lah, miss.
Alright, so good luck in your UPS 1 later. Just stay calm. You will be able to do well. So